Hi, I'm Kai. I'm a researcher at the Department of Diagnostic Science at UZ and I'm establishing a culture method to grow blood stem cells from induced pluripotent stem cells. Our bodies are made up of billions of tiny building blocks called cells. Each human being develops from one fertilized egg cells that grows into an embryo and a fetus. Therefore, the earliest cells that are formed within the first days after conception are so-called pluripotent stem cells, the mothers of all cells that have the potential to give rise to all different types of cells that make up our body. Throughout development, these pluripotent stem cells give rise to more dedicated stem cells that have the potential to develop into limited but not all different types of cells. For example, heart stem cells can give rise to the different types of muscle cells in the heart that contract and pump the blood around the body, but no longer to intestinal cells or others. Ultimately, these dedicated stem cells contribute to the development of all organs in our body that are unique and have specific functions. During our lifespan, that can give rise to new specialized cells to replace the old or damaged cells in order to maintain the function of our organs. This also applies to our blood. In our bone marrow, we have blood stem cells. Blood stem cells are dedicated stem cells that produce a wide range of specialized cells found in the blood. For example, blood cells that carry oxygen and different types of white blood cells that protect us from infections. The proper functioning of these blood stem cells is crucial, as our blood cells are quickly renewed and our blood stem cells have to ensure that there are enough of all the different types of blood cells. If something goes wrong in those blood stem cells, such as errors in their genetic material, these can lead to blood diseases and cancers like leukemias. Currently, patients with leukemia and other blood diseases can be cured by a blood stem cell transplant. In this case, healthy blood stem cells from a donor are given to the patients to replace their abnormal blood stem cells. However, successful transplantation requires that the patient and the donor mesh immunologically. Otherwise, patients will recognize the donor cells as foreign and reject them. Immunomatching donors are rare and the search is often time-consuming. Therefore, it will be a huge advantage to have a way to make healthy blood stem cells from the patient's own cells, and via iPSCs, this might actually work. With the advance of science, researchers found ways to reprogram specialized cells back to a more stem state, as such making induced pluripotent stem cells, or what we call iPSCs. Growing these iPSCs into the cell type we want depends on the addition of specific growth factors and nutrients. While these turn out to be a very effective technique to make cells like heart stem cells, scientists are yet to figure out the correct recipe to grow blood stem cells from iPSCs. In our lab, we now found new information that might be the key to finally derive blood stem cells from iPSCs. With the support of CRIG, that is exactly what we will further investigate in this project.